Okay, welcome back to the Classics in Immunology Journal Club. Before we get started, let me just remind you to like the video if you do, subscribe, then click on the bell, get notified for when there's another. And also check out my website, and the writing tab should have all the classic papers of immunology that we've already reviewed since 1798 and Edward Jenner, the first vaccine against smallpox. And that's appropriate because today we're going to talk about another vaccine against coronavirus. The paper for today is by Jinyan Liu, Abishek, Chandrashekar, David Sellers, 8 additional authors, and Dan Barouch, Beth Israel at Harvard. And the title of this paper is Vaccines Elicit Highly Cross-Reactive Cellular Immunity to the SARS-CoV-2 Omicron variant. And it came out in Nature on St. Patrick's Day, March 17th, 2022. So we're in basically as real time as you can get, essentially, in terms of what scientific articles are all about. Now, the background on this paper is, I think it's important to realize and to point out that until now, really, essentially, all vaccines that have been generated and produced and used were tested for their ability not to prevent infection, but to prevent the disease, whatever it was, measles, mumps, chickenpox, smallpox, and which was recognizable by the symptoms of those diseases. And if you think about it a second, I mean, that makes sense because back in the day, Jenner, he didn't have a test for smallpox virus. People didn't even know what viruses were at that point in time. They tested for the pox because they could see that on the skin surface. And that was basically a delayed type hypersensitivity reaction in the skin to the presence of the virus. And as one of the things that I've pointed out in previous episodes of the Journal Club, the symptoms of most infectious diseases, contagious diseases, are not due to the bug. The bug is not making toxins and poisons and all kinds of bad molecules and producing them to make all the bad symptoms of the disease. For the most part, the symptoms are caused by the immune reaction to the bug. And so a good immune reaction is for the immune system to clear the bug, to recognize it and respond to it, detect it, and get rid of it before the whole immune response really revs up and becomes manifest, essentially, by the symptoms of inflammation. So those are the things, and that was used for the initial vaccines that were tested for COVID. They didn't test for the virus because they were immunizing thousands and thousands and thousands of people. They told the people to call us if you have symptoms. That's the way they determined whether or not their vaccine worked. And so the results were quite fantastic because I've never seen vaccine results as good as they got from the messenger RNA vaccines that were used in the United States, produced by Moderna on the one hand and Pfizer-BioNTech on the other. It was only after HIV in the 80s and 90s and aughts and so forth that the molecular virologists got involved, identified all the genes and the gene products of the virus, identified the receptor on the cell surface of the T cells that the virus infected. There were more than one receptor for the virus. And that, for the first time, allowed the generation of tests for the virus. Now we have a point of care test for coronavirus, and that tests for a coronavirus protein antigen in the nasal swab for that whole thing. So that's sort of the background with the looming of the new of a whole pandemic that was happening. The molecular virologists in the case of the coronavirus said, they had recognized what the spike protein was the major protein that was binding to the cell surface, and it was binding to the cell surface through a receptor binding domain. And so that they could make a vaccine from the spike protein that would activate the immune system to make neutralizing antibodies that would prevent then the spike protein from binding to the cell surface and therefore prevent infection. This was a whole new ballgame in terms of vaccinology. But as soon as they announced that this is what was going to happen and this is what they were doing and this is what they've been working on, I said to myself, well, that's an awfully specific vaccine. What happens if the virus mutates and escapes from those neutralizing antibodies? And of course, that's what happened. And that's what's given us several variants, in particular, the Omicron variant that has escaped because it's gotten around the neutralizing antibody raised by the initial vaccines that were raised against the spike protein genetic sequence from the initial isolate from China, from Wuhan. Now, the other thing that is interesting to point out in this regard is the fact that amongst the vaccinologists, and as a consequence of the difficulty of generating a vaccine for HIV, there's been a sort of an undercurrent of a battle going on between the humoral vaccinologists and the cell-mediated immune vaccinologists as to which aspect of the immune response is important for protection against whatever the disease is. HIV was the one that generated, I think, the most recent controversy. So that's the background of this paper. And so the question is, okay, we're probably going to have additional so-called variants. I prefer to call them mutants that escape even the new vaccines, the so-called bivalent vaccines against the Omicron variants. In the introduction of this paper, they point out that the recent studies have shown that the vaccine elicited neutralizing antibodies to the initial sequences of the Wuhan variety were markedly decreased in terms of the vaccine efficacy against the Omicron variants as they arose. And so the question that they approached in this paper was, what's the status of the cell-mediated immunity in these vaccine recipients that the Omicron has escaped from the neutralizing antibodies? And so they addressed both CD8 positive, the immune responses, as well as CD4 positive T cell immune responses in subjects vaccinated with two different coronavirus vaccines. One that was helped to be developed by Dan Barouch in his laboratory at the Beth Israel in Boston, that was ultimately then produced by Johnson & Johnson with the adenovirus as a vector to bring the spike protein into the cells of the host, of the vaccinated individual. And that the results from that vaccine were then compared with the Pfizer-BioNTech vaccine. And so, this is the first time, at least that I'm aware of, that this head-to-head comparison of one vaccine from one company, one outfit versus another one, because, this is capitalism we're dealing with, and no company wants to go head-to-head with another company's vaccine in case they lose. And therefore, all of their work would be for naught, and it wouldn't make as much money. So this makes this paper unique for two reasons. One is they carefully looked at the cell-mediated immune responses in these vaccinated people, and they compared two different vaccines head-to-head. That's good. That's why that's in addition to the fact that their results were absolutely striking. That's why this paper is a major article in Nature. So let's go to the screen sharing and see what they found. So this is figure one from the screen sharing. And in the A panel at the top, what we have is neutralizing antibody responses. And on the left-hand portion, we have neutralizing antibody responses in response to the adenovirus, the Johnson & Johnson vaccine. And in the right-hand panel, we have the Pfizer-BioNTech vaccine. And then we have the antibody responses against the spike protein of the Wuhan variety, the Delta variant, the Beta, and the Omicron variant. So there's Wuhan, Delta, Beta, the Omicron variant here. After 1 month after the initial vaccination, and then 8 months after the initial vaccination. And by and large, what you can see is that the responses at 1 month were pretty good against the immunizing or the initial variant, or the initial virus. But the Omicron has escaped from that. Both at 1 month after the vaccine, and also 8 months later, it hasn't gotten any better. Same story with the BioNTech in terms of the Wuhan variant, or the Wuhan virus, and then the Omicron variants. In the B panel down here, this was a neutralizing antibody response. And in the B panel, we have an ELISA assay looking at the receptor binding domain of antibodies in the sera of individuals and against the receptor binding domain of Delta, Beta, and Omicron. And you see basically by ELISA and simply binding to these different proteins, the fall off of the Omicron. Same with the BioNTech messenger RNA vaccine immunization. So then if you go on to figure 2, so that's what basically we already knew that. But if you look at figure 2, get down here. If you look at the cell-mediated immune responses to the Omicron, you can just glance at these figures and look at the Omicron data right here, compare that to the other variants and so forth, and you've got good cell-mediated immune responses, both at 1 month and at 8 months. This is after adenovirus vaccine. This is after, and this is ELISpots in the A panel, and down here, or this is CD8 responses, and these are CD4 responses. And so it's clear that the cell-mediated immune responses are holding up quite nicely. By comparison, and this is before vaccination, these are just the negative controls here. These are CD8 cells here, there's CD4 cells here. And so the bottom line is that the cell-mediated immune responses measured by both ELISpot and also by intracellular cytokine staining are quite robust after both vaccinations. And so these responses were tested after with using overlapping peptides in the entire sequence of the spike protein, so that they were 16 amino acids long with 11 amino acid overlaps. And so that's major. It's one of the major differences between a humoral immune response and a cell-mediated immune response. There's simply more antigens, more epitopes, more peptides that the immune system is recognizing. And so I'm going to stop there and stop the screen share and go back now. And I urge everyone to go and read this paper. And there's a lot more. There's more data that I won't go over this today, because the major point is there are great cell-mediated immune responses in this infection. And so if you look at the discussion of this paper, they say that in the very first sentence or two, they say that the data clearly demonstrate that both adenovirus and BioNTech vaccines elicit broadly cross-reactive cell-mediated immunity against Omicron, the Omicron variant. And these immune reactions, the extensive cross-reactivity between the Omicron contrasts sharply with the decrease in the neutralizing antibody. That's what basically allowed the president of the United States to say the pandemic is over, because What the cell-mediated immune response protects against is the disease, serious disease. People getting sick enough to have to go to the hospital, sick enough to be placed into the intensive care unit on a ventilator, and sick enough to die. That doesn't happen. And it doesn't happen because of the cell-mediated immune response. Now, you say to yourself, how does cell-mediated immunity do that? And I think there are two. Basically, it's a whole other aspect of things, but they mentioned this in the discussion of this paper. Is one of the major mechanisms that cell-mediated immunity has is cytokine production. Cytokines, particularly interferon gamma, because if a vaccinated person who no longer has neutralizing antibody effectiveness against a new variant like Omicron, but does have cell-mediated immunity against the spike protein, multiple peptides in the spike protein, then if that virus rears its ugly head in the community and the person becomes infected with that virus because it doesn't have neutralizing antibodies, it does have T cells that will recognize that virus. And those T cells will make interferon gamma. Interferon gamma is essentially, its initial designation or name was macrophage activating factor. That activates the macrophages to essentially do all the good things that macrophages do. And the interferon gamma also activates all the other cells that have an antiviral effect. So they take the place of the initial innate immune response and the interferon alpha and beta that prevent the widespread replication and infection in this kind of instance. And of course, the other thing that cell-mediated immunity does is that the killer T cells kill virus-infected cells, stopping the spread, and ultimately ridding the host, ridding the person of those viral particles. And so now what we should do, so that's the essence of the discussion in this paper. So the question is, what should we do now? And then this is what we probably what should have been done back in the very beginning. It was very logical to try to stop the spread of the pandemic by preventing infection with the very specific spike protein vaccines. But now we should be using the same technology, the adenovirus and the messenger RNA or whatever, using different gene products from the coronavirus that are the primary ones that are expressed on virus-infected cells, which now they can find out about, and I'm sure everybody's looking for those, the ones that are primarily expressed, because you should be able to produce a vaccine that would prevent all coronavirus viruses and variants and mutants and from causing serious disease. And so you might get the sniffles for 1 or 2 days or whatever, but you won't have to go on a ventilator. And so that's sort of the bottom line, my bottom line in this paper. And I think this is an extremely important paper for the whole pandemic, and everybody should read it carefully. So I'll stop there. Don't forget to like the video and click on the bell and get notified for more. Subscribe and check out my website. Check out the writing tab. Read this paper. So thanks very much for coming. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.